So all of those questions that we were just asking were all good and well and everything like that, but in order to study those questions and ask those questions, you have to have a way to actually get that protein, to purify that protein. So studying proteins and peptides sometimes requires some purification from a mixture. Polypeptides contain differing amino acid sequence. The sequence and arrangement of amino acids gives the polypeptide some chemical character. It can tell you about how hydrophobic it is, how polar it is, what the overall charge is. Some polypeptides bind to specific targets, which can be used to kind of collect them or fish them out of a complex. So if you have you have a protein that's positively charged, do you know what you would need to do in order to separate from the rest of its mixture? Yeah, find something or use a bait basically that's negatively charged. Now, a mixture of proteins can be separated by a number of different physical and chemical properties. You can use the charge of the protein, the size of the protein, the affinity for a ligand, the solubility, hydrophobicity, and thermal stability. Now, all of these are properties that can arguably be exploited. Chromatography, much like what you did in your organic lab, chromatography is commonly used for preparative separation in which the folded protein is able to be kind of fished out. So chromatography that you did in organic is similar, but it's a little bit different. Column chromatography is what is kind of the name of the game, much like you probably did in organic. But what makes this a little bit different is what are those small beads? In your organic lab, you probably did silica gel chromatography, or you use silica as your resin for selecting things. In column chromatography, where you're separating proteins, you're going to have some modifications to that. So what this shows is your simple schematic. You've got 3 different substances, substance A, B, and C, and one of those substances is basically going to get stuck into that column or stuck in that column for a longer period of time. You're going to have some means of detecting something, which we have, we'll get into this in a little while, but we have ways of detecting proteins by using their absorbance at 280 nanometers to say whether or not protein is in a sample. So you'll have to have some sort of detection you'll have to have some sort of detection and recording. So then you can say, okay, well, absorbance at 280 nanometers, fraction number 1, 2, 3, 4, fraction number 1 and 2, they had 0 absorbance at 280, but 3 and 4, those had values. What that tells you, if they have absorbance at 280 nanometers, so that tells you that there is protein in there. Is it your protein? No, not necessarily, but there is protein in there. Okay, so ion exchange chromatography is the first chromatography technique that is, I'd say it's the most easily understood. Excuse me, the reason for that is you're using nothing but charge to separate your protein from the rest of a mixture. What you're going to do is you're going to say, okay, well, I have positively charged proteins. Those positively charged proteins, I know they will bind to something negatively charged. Okay, so if I have a medium or a resin that is negatively charged and I have positively charged proteins, well, those positive charges on that protein will adhere to the negative charges on my resin, and they will stay there indefinitely, or maybe not indefinitely, but they will stay there for a while. Compare that to negatively charged proteins. So we've got our negatively charged proteins in our 2 different colors. We've got red and kind of a light red. Those very negatively charged proteins, they have the least affinity for binding to this resin. And so those are going to come out of this column earliest. They're going to come out in fractions number 3, 4, 5. But then later on, those proteins that have a little bit less of an overall negative charge are going to elute from the column. Size exclusion chromatography, also known as gel filtration chromatography. This is also kind of simple to understand. This is a chromatography technique where you're separating based on size. And it's as simple as The largest proteins elute first. The smallest proteins elute last. So if you have a mixture, large proteins, what they are going to do is they are going to migrate through that column very quickly, whereas the small proteins are going to migrate very slowly. We've got this cutout right here, and basically all of the beads that make up that resin, they have very small pores in them. The way I kind of think about it is you've got a solo cup, and this is the example that I've been using for years and years and years. If you had a baseball and you tried to throw that baseball through a solo cup, what's going to happen? It's going to get stuck, and if you put a little bit of force behind it, it will make its way through, compared to a basketball. If you threw a basketball and you tried to make it through a solo cup, what's going to happen? Well, it's going to go to the entry, and then it's not even going to make it through, because it's never going to enter it. So that's the key defining factor. Does the protein enter the pore within the bead? The last separation technique is affinity chromatography, or the last chromatography technique is affinity chromatography, and that's going to use a ligand. So let's say that you have glucose, and you take these beads and you add glucose to them. The glucose is simply an example, but we make that our ligand. Well, what's that going to do? That's going to bind any proteins that have a high affinity for glucose. What's it going to do to proteins that have no affinity for glucose? Nothing. Those proteins are going to float straight through. So what you're going to be able to do is get glucose to serve as the bait, and then your prey is going to be those proteins that bind glucose. They're going to bind and they're going to adhere. They're going to adhere until you do something that causes the proteins that have a high affinity for glucose to release. One way that you could do that is you could use free glucose, or you could use another hexose that has a high affinity for your protein that's bound to glucose. So electrophoresis of proteins is a good analytical technique. So maybe if you've taken a genetics class, you've done DNA gel electrophoresis. Electrophoresis uses an electric field and pulls proteins according to their charge. The gel matrix hinders mobility of proteins according to their size and shape. The gel is commonly polyacrylamide. Separations of proteins via electrophoresis is often called polyacrylamide gel electrophoresis. So here's an example of an SDS-PAGE. This is generally going to be a vertical gel. You're going to load your protein sample at the top. You've got your negatively charged anode. You've got your positively charged cathode. You are going to send your proteins towards this positive charge. You're going to send an electric current through there. And what's going to happen is those proteins are going to separate based on charge. SDS-PAGE separates based on molecular weight. It also separates by charge, but the reason that you're able to say it separates by molecular weight is because we use this SDS, sodium dodecyl sulfate. This is going to basically line the protein and give the protein a uniform negative charge. So I'm going to say, yeah, here we go. SDS gives all proteins a uniformly negative charge. So you're not separating a positively charged protein from a negatively charged protein. Every single protein, regardless of what it looks like, and regardless of the composition, because of SDS, you are giving negative charges across the board. So then what you have is the smaller proteins have fewer negative charges applied to them compared to a larger protein. Now what that means is the rate of movement will only depend on the size. So small proteins move faster. Now one thing that I'd like to say about this that makes SDS-PAGE different from chromatography is SDS-PAGE is an analytical technique where you're able to analyze the molecules. You're not trying to separate them. You're separating them, but you're not, and this might sound a little bit weird, but you're going to separate, but you're not going to be using them. So this is a technique used for analysis rather than kind of a preparative technique. So here's an example of an SDS-PAGE and what it looks like. So what we've got are induced cells. So this is E. coli. This would be an example of an E. coli protein expression where you're just growing cells and hoping your protein gets expressed. The uninduced cells are you've done nothing to tell that cell, hey, start producing my protein. Excuse me. The induced cells are samples in which you have added something to tell the cells, hey, start producing my protein. Okay, so that's what we see this band right here. This band, as you'll notice, is not in this lane right here. Okay, so we're going to look at those induced cells, and that's the sample that we're going to proceed with. We're going to look at a soluble crude extract. So this shows us all of our soluble proteins, and I'm drawing a poorly straight line. Yeah, not a very good straight line. Those are all of our soluble proteins. The next sample is your next or your first sort of preparative technique, and that's ammonium sulfate precipitation. In this, we did not go into it, so you didn't miss out on anything there. This is something that I'll talk about if you end up taking Chem 3276, which is the lab component of this class. It's called experimental biochemistry. And what we need to be concerned with, though, are these right here. When you separate this protein with an anion exchange, a cation exchange, or a cation exchange, what you're able to do is you're actually able to collect that protein. So since this protein was able to adhere to an anion exchange column and a cation exchange column, does that tell you that that protein is positively or negatively charged? No, instead what it tells you is that it's simultaneously got positive charges. Hence, it's going to bind to the negatively charged or the negatively charged resin of the cation exchange column. It also has negative charges on it, enabling it to bind to the positive charges on the negative charges of the anion exchange residue. And that does seem a little bit weird, but think about it like this. A positive charge on a protein, a cation, is going to bind to and stick to a cation exchange column. A negative charge is going to bind to and stick to an anion exchange column. SDS-PAGE ultimately can be used to calculate molecular weight of a protein, and that gets us to this point right here where we are looking at a protein gel. SDS-PAGE can be used to calculate the molecular weight of a protein, because what we're going to do is we're going to use all of these different molecular weight standards, and we're going to say, okay, well, our largest protein in this molecular weight standard migrated from here to here. We know that this protein is 200,000 Daltons. Our next protein migrated from there to there. It's approximately 116,000 Daltons. The next one, as we move down, ultimately we're going to get to ovalbumin as an example that has migrated from the top of the well down to here and is approximately 45,000 Daltons. And the one below that is carbonic anhydrase, which also moved from the top of the well down to here. That is 31,000 Daltons. Our protein right here, the one that we are curious about, migrated from the top of the well down to here. What can we say about it? Well, we know that it's larger than 31,000 Daltons. We know that it's smaller than 45,000 Daltons. But what we can do is we can take a log of the molecular weight standards, all of these guys right here, and graph them against their migration distance, and ultimately come up with a linear regression curve and say, well, if it migrated this far, well, then it has to be this exact molecular weight. So this is one way that SDS-PAGE, or the data that you get from SDS-PAGE, is very valuable.